The reason that we discuss these systems of classification, these methodologies by which we kind of map this evolutionary relationships or these evolutionary relationships, um, particularly with respect to physical anthropology, comes down to how on earth we place fossil hominins, fossil non-human primates into the kind of existing framework or existing classification system that we have for extant species. And so we've got to recognize how to interpret new fossil finds. I mean, it was one thing in this kind of era of European discovery, which is the positive spin on exploration, imperialism, and colonialism. It was one thing to figure out how newly contacted species or newly observed species, at least new to Europeans, fit into kind of this greater arc of biological diversity. But it's entirely different to try to place these fossil forms. When we look at the fossil record, we've only got about 5% of the diversity that the Earth has ever held. So 95% of both species and specimens never end up fossilized. So we've got very few species that are present in the fossil record. For some of these species, we only have one specimen, or we've got an overabundance of specimens of a particular age and sex class, but fewer, let's say, at perhaps more important ages for particularly for determining or inferring kind of the pace of development or life histories. So we do get, for example, adult both males and females, but we have very few fossilized infants. We have very few fossilized toddlers. So all of this leads to interpreting difficulties. We have to allow for individual variation, right? I mean, we recognize just even looking at skeletons and skulls from modern Homo sapiens that we've got an extreme level of individual variation. Um, we've got, for example, the difference in height between individuals affected by the genetic factors that cause dwarfism versus those that have something like Marfan syndrome, right? We've got individuals who are tall, individuals who are short, individuals who have very kind of stocky build and thick frames versus those who are more long and lanky. So it's hard when we're basing our species designation or species kind of typology on only one or two specimens to recognize what are going to be greater like species level differences or genera level differences versus what's just individual variation. We also have ontogenetic variation. This is age related variation. So for example, we change in height over our period of growth and development. So if we've got a skeleton that has short stature, how do we infer whether they were a short statured adult or whether they just weren't done with growing? I mean, we can look at things like whether the epiphyses of the long bones are fused or not, but if we're only looking at bone fragments, we may not have a complete picture. We can look at dental eruption patterns and infer some things about age based on the order and age at which permanent dentition comes in versus the deciduous dentition. But what if we only have post-cranial skeletons? What if we don't have the skull or particularly the jaw? We also see a large degree of sexual dimorphism, and we recognize this as a trait across primates that terrestrial species, like gorillas, like baboons, like some species of macaques, have greater body size sexual dimorphism. This is variation across the sexes. And generally, when we've got sexually dimorphic species, we've got males on the larger end and females on the smaller end. So if we allow for all of this level of variation, the individual variation, the ontogenetic variation, and the possibility of sexual dimorphism, then how on earth do we determine when we should draw species designations, where we should kind of draw the line between species? This brings up two terms. There are paleoanthropologists, and this is true across paleontology, not just paleoanthropology, but there's a school of thought of people who are called lumpers, and there's those who are called lumpers are those that tend to perhaps even overrepresent um, polytypic representatives of the same species. And so lumpers would tend to take a large number of fossil finds from a vast region, particularly those that date to, certain time periods, and include them as polytypic individuals from within the same species designation. Splitters, by contrast, are apt to find any kind of variation that we see present, whether it's individual variation, whether it's sexual dimorphism, and combine those together in the same, divide those apart, use those as the basis for drawing species lines. And so we end up then with a plethora of new species named. I mean, there's something to be said for, there's a draw within paleoanthropology, for example, to have species named after you. I mean, that's considered one of the biggest kinds of finds, or biggest ways to make your name is to discover a new quote unquote species and then name that new species. But we have to recognize that as we're looking at the paleo record, we've got spatial variation. I mean, and this is reflective or indicative of environmental, kind of smaller scale environmental variation, but we also have temporal variation. And so we've got to somehow come up with classification schemes that allow for and accommodate both this spatial and temporal variation. Grouping fossil finds into species is certainly a hurdle that we have to cross, but grouping fossils into genera is particularly problematic. I mean, we recognize that when we look at modern species, that we've got this hybridization issue. We can comment on these hybrid species, or the degree to which they are able to produce these viable fertile offspring. And this, when species are able to hybridize, this helps us to determine that they are in fact members of the same genus, but we can't infer those kinds of things from fossil finds. So with fossils, we're looking for, instead of specific ecological niches that are occupied, we are looking for the same broad adaptive zone. This is a more general ecological lifestyle that is kind of applicable across wider contexts, both spatially and temporally. So we look for these very specific ecological niches for species designations, but more broad adaptive zones for genus designations. And so as we're looking at something like early fossil hominins, this is this and the lumper versus splitter kind of context or argument comes into play as we're looking across Australopiths, for example. Some naming schemes have all Australopiths under the genus Australopithecus. Some naming schemes separate out the robust Australopiths into their own genus called Paranthropus, because of these highly specialized adaptations for this kind of raw, thick, fibrous vegetarian diet. And that's associated then with some of these anatomical changes like the sagittal crest, like this massive lower jaw, so on and so forth. So it's particularly problematic when we're interpreting species and genera in the fossil record. It's particularly problematic, like how we define this broad adaptive zone. The process of fossilization predisposes some species, some locations to yielding a wealth of fossils versus others that really don't provide us any fossils at all. Fossils are trace organisms, um, or traces of ancient organisms. Fossils include hard tissue. Many, in fact, are comprised of hard tissue. Hard tissue being parts of the bodies, parts of the specimens that were already made from minerals. This can include teeth, this can include bones, this can include eggshells, etc. Mineralization is the process in which parts of the animals become transformed into stone-like structures. And so mineralization is more common for parts of the body that already yield or that are already partly mineralized. Diagenesis is the chemical exchange between the minerals of a dead organism and its surrounding burial environment. So this gives us some insight into or ideas of the methods by which fossils are generated. There are also other processes by which fossilization can occur. Part of it depends on how an organism dies, and part of it depends on where it dies. Taphonomy is the study of how bones and other materials can be preserved as fossils. So we all recognize, for example, um, the kind of mythology of Jurassic Park, for example. In Jurassic Park, of course, we had the mosquito that bit a dinosaur and then was preserved in amber. And by sequencing the dinosaur DNA that was present in the blood and the digestive tract of the mosquito, we were thusly able to bring dinosaurs back to life. So there are certainly insects that are preserved in things like tree resin, amber being one of those. There are, of course, e- even things like feathers of dinosaurs now that have been preserved in tree resin that have helped to answer this question on the origins of bird species. And so resin is certainly, tree sap is certainly a way that, particularly even some soft tissue can fossilize. We also see this process of the fossilization of impressions, and so footprints, for example, can rapidly fill with sediment. Um, and thusly be preserved. And we've got some famous early hominin footprints, the Laetoli footprints, that give us some insight into when bipedality evolved um, for early Australopiths. So the process of fossilization then depends on these prevailing ecological characteristics. There's some areas that the Africa's Rift Valley, for example, has an abundance of fossils. Well, it was a pretty dry area. It had pretty remarkable, um, the things that died didn't decompose as quickly as they would in like dense tropical rainforest, for example. So the aridity of an ecosystem can affect the process of fossilization. Sometimes the preservation can affect fossilization. And so, we've got, for example, mummification, which um, can help to preserve specimens so that they are more likely to last through time and to either fossilize or perhaps just be preserved. When we look at the history of life on Earth, we are going to talk about certain clades that we are particularly interested in. Primates are mammals, and at a higher level than that, they are vertebrates. And so we're going to do just a brief history of vertebrate evolution. When we talk about evolutionary time, we're talking about this geological time scale. This is the organization of the Earth's history into eras, periods, and epochs. We've got our earliest vertebrate fossils that appear early in what's called the Paleozoic, around 500 million years ago. During the Paleozoic, fish, amphibians, and reptiles abound. By about 250 million years ago, we have the appearance of what we call mammal-like reptiles, like Dimetrodon, that then end up giving rise to a diversity of early mammals. Many of these early mammals were then killed out in or ex, driven to extinction by a mass extinction event at the end of the Paleozoic. Vertebrate evolution has been affected or influenced by geographical events. This can include continental drift. By the end of the Mesozoic, continents were beginning to assume their current positions. This resulted in um, a large-scale kind of geographic isolation, particularly of species in places like South America and Australia. I mean, why do we have modern-day marsupials in Australia? It's because Australia moved farther away from the rest of Gondwana land, or these southern hemisphere continents. And thusly, the marsupials that were present were geographically isolated. Similarly, there are some key differences between what we call New World species and Old World species. Once South America diverged from the rest of Gondwana land, we end up with geographic isolation. Many of these species can't cross the ever increasing kind of ocean expanse. So long-term drift profoundly isolated groups of animals, and this was particularly marked with land vertebrates. I mean, those that can fly, those that can swim are perhaps less affected by the movements of continents. The Mesozoic was this era that was mostly reptiles. This includes the diversification of dinosaur groups. The Cenozoic then follows another mass extinction event during which the dinosaurs were wiped out, and this left ecological niches open for mammal and bird radiations. We divide the eras, or the Cenozoic year, into periods, including the Tertiary period and the Quaternary period, and we divide then those periods into epochs. And so, as we move forward and talk about primate evolution, we will look to these different epochs, including the Paleocene, the Eocene, the Oligocene, the Miocene, and the Pliocene. We will also, in the Quaternary period, talk about the Pleistocene and the Holocene. And so, as we look to discussing primate evolution, we'll talk about the Paleocene being this period of early mammal radiation, the period during which the first primates evolve. We'll talk about the Eocene being this kind of age of prosimians, where prosimians diversify and then the first anthropoids appear. We'll talk about the Oligocene being this period of, epoch of proto-monkeys and this diversification of monkeys, separation of catarrhines and platyrrhines. We'll talk about the appearance of the first proto-apes at the end of the Oligocene. The Miocene will be kind of the planet of the apes, this period of rapid ape diversification. But at the end of the Miocene, we get our first fossil hominins. The Pliocene is the proliferation or radiation of Australopiths and the appearance of the first members of the genus Homo. The Pleistocene being the diversification of the genus Homo, the extinction of robust Australopiths, the appearance of the first members of our own species, Homo sapiens, and then the Holocene, this massive diversification of humanity, the spread of modern Homo sapiens across the rest of the globe. When we talk about the evolution of mammals, we've got to look to factors that contribute to their adaptive radiation. I mean, mammals wouldn't have adaptively radiated if dinosaurs had not been wiped out. Dinosaurs kind of maintained only these small nocturnal populations of mammals because they would have been prey species to many of the larger bodied dinosaurs that were present. Mammals, compared to reptiles, have a larger brain that's better equipped to process information. This is particularly notable in their cerebrum. The cerebrum is the fore brain, and the cerebrum includes the neocortex. Not only did we increase the size of the cerebrum, including the neocortex, but we also started to generate these convolutions. We started to fold the brain in on itself. What this does is, without having to include the volume of the brain, this includes the surface area of the brain, giving more sites for neuronal proliferation. So by increasing the surface area, we increase the number of neurons we can pack into the brain, which increases our ability to process information. Mammals have a longer, more intense period of growth. This is afforded by both internal fertilization as well as internal development. And so when we look at the differences, for example, between monotremes versus marsupial mammals versus placental mammals, monotremes are kind of limited in the pace at which the embryo can grow and how hard it can, or how large it can grow, by the nutrients that are available in the yolk. With marsupial mammals, there's a really high, what we would consider kind of neonatal um, death rate because they're born at such a primitive stage, or such a less differentiated, smaller stage of development, and they have to make that long trek from the birth canal all the way up to the teat, and kind of nestle in the mother's sac to continue the rest of their development outside of the uterus. Placental mammals have a distinct advantage in that they are able to have a more efficient transfer of nutrients to the developing organism, which means that they can be born at a kind of farther along stage of development, which means that they've got an advantage in terms of both processing information, and they're able to give birth to larger brained infants, for example. But they also have an advantage in kind of the degree of altricialness versus precociousness. Altricial infants are very helpless, rely a lot on mom, are very prone to neonatal mortality. Precocial infants are less likely to die at younger ages. They're able to fend more for themselves. And so, we can look across taxa and see a difference in litter size and precocialness. Generally, animals that are born in litters are more helpless when they're born because they're born at an earlier stage of development, whereas those that are singleton births are more, let's say, vigorous, or further along in their development, and better able to regulate their own body temperature. Of course, we've got cases like giraffes, zebras, horses, etc., where they're able to run and keep up with the herd within hours after birth. Another advantage that mammals have over reptiles is a heterodont dentition. Heterodontia is having different kinds of teeth. When we look at reptilian mouths, many of their teeth have the exact same shape, whereas we look at mammalian teeth and we've got differentiation between incisors, canines, premolars, and molars. What this allows is for teeth to be used for different functions and for teeth to be used for different resources. So reptilian mouths are really geared towards kind of one particular diet or dietary specialization, whereas we are able to see mammals with more omnivorous diets because teeth can be used for different functions. So we're able to process a wider variety of foods. This will relate to kind of adaptive specialization. The last factor that contributes to the success of mammals in our adaptive radiation is being endothermic. This is warm-blooded or maintaining a constant body temperature, and this is true, of course, of theropod dinosaurs that then evolve into birds as well. What this allows is the maintenance of a high metabolism kind of around the clock versus having to change your behavior based on the need to either warm or cool your body down. And so, of course, when we have periods of extreme cold, reptiles kind of go into torpor or hibernation, right, where they slow their metabolic rate down so that they can survive these times of extreme cold. We are able to still maintain a fast metabolism across a wide variety of temperatures. So we don't have to stay in tropical areas, for example. We are able to still maintain high levels of activity, given, of course, or provided, of course, supply of food, even in more temperate and then colder areas. All right, so we've got several processes that that work together to kind of culminate in macroevolution. One of these processes is the adaptive radiation. This is the relatively rapid expansion and diversification of life forms into new ecological niches. Generally, a species or a group of species will diverge into as many variations as two factors allow. One of those factors is the species' adaptive potential. Another one of those factors is the adaptive opportunities of the available niches. So if there are a wide variety of niches available and we have a kind of generalized kind of quote unquote primitive ancestor species, that ancestor species can specialize, which relates to the second point as well, this transition from generalized to specialized traits. That ancestor species can radiate into a variety of ecological niches. If we only have one environment that's available, we don't have a lot of differentiation, we don't have a lot of different adaptive potentials, then we're just going to see one more generalized form of the species. As we relate to this transition from generalized to specialized traits, we do not see specialists evolve into multiple species of specialists. Rather, when we look to these ancestor species, they all have more generalized traits. Generalized ancestors provide the flexible evolutionary basis for rapid diversification. And so these specialized characteristics are nearly always derived. So when we think about kind of species that provide the opportunity for further differentiation. We're not going to see, for example, specialists like koala bears or giant pandas evolve into even new species. I mean, their diets are already way too far specialized. I mean, people have a mythology, for example, of Planet of the Apes. This idea that we will be able to give an injection of human DNA to chimpanzees, and then all of a sudden they will walk upright, wear clothing, speak in complete sentences. Chimpanzees are highly specialized apes for their specific ecological niche, and they're going to be able to some degree, change with differing ecological changes, but they're never going to evolve into humans. We shared a much more generalized ancestor, and these different ecological niches allowed for the specialization between gorillas, chimpanzees, and humans. Gorillas are highly specialized for forest dwelling, highly specialized for eating copious amounts of vegetation, right? This, kind of plant stems, leaves, etc. material. Chimpanzees are also specialized for the forest, but more oriented towards diets high in fruit. Hominins became specialized for life in the savanna, and that required a completely different suite of resources. So chimpanzees are too specialized for forest living to be able to effectively survive solely on savanna-like resources. So, we have to recognize that continued paths along this kind of continuum towards more specialized traits leads to evolutionary dead ends ultimately, right? Leads to the inability then to further adaptively radiate. And so we've got debate as to whether there is a difference in the processes that give rise to macro versus microevolution. we used to, even Darwin himself thought of microevolution and macroevolution as kind of two ends of the continuum. About 40 years ago or so, there was further interest among evolutionary biologists to really examine the processes of micro versus macroevolution, maybe perhaps to drive a wedge in between them, to determine how they were different, but that hasn't really borne to fruition. It's generated a lot of hypotheses. Those hypotheses then have not stood up to rigorous testing. So we don't find, for example, a lot of differences between the mechanisms of micro and macroevolution. Rather, the major difference appears to be one of scale, that they exist at two ends of the same spectrum. Natural selection is influenced by how fast the environment is changing and how fast genetic changes appear and spread within a species. So, this role of the environment in kind of setting the pace of evolutionary adaptation is of key importance. We also see a difference across members of the same lineage in the pace of their evolutionary change. The pace of evolutionary change can speed up or slow down due to the factors that influence the size and isolation of populations. So there are some groups that simply have faster or slower rates of speciation. This is true across primates, as well as other taxa. For example, Old World monkeys speciate a lot more slowly than do great apes. And so we've seen a lot more constancy in the fossil history of Old World monkeys. We've seen less radiation of Old World monkeys versus this Planet of the Apes that we saw during the Miocene, where we saw very rapid ape differentiation. It's this rapid differentiation or speciation of apes that really kind of predisposed the hominin line to diversification and speciation as well. If we hadn't evolved from our proto-ape, proto-human ancestor, if we had a more ancient branch from Old World monkeys separate from apes, then we may not have adapted so rapidly to the widening savanna. We might be much more baboon-like, or share our ecology with baboons. So we don't need to consider microevolution and macroevolution as separate entities. Rather, the same kind of processes govern microevolution at a shorter time scale and result in macroevolution over a lengthening time scale. They represent two potential outcomes if we just kind of widen our time of focus. And so in summary, recognize these two major organizing perspectives to understanding large-scale evolutionary history of life on Earth. This includes taxonomy, whether that taxonomy is evolutionary systematics or whether that taxonomy is cladistics, and then this geological time scale. This geological time scale gives us the depth and the length of time over which evolutionary change can happen. Understand and recognize these two different approaches to classifying and interpreting life forms, including evolutionary systematics and cladistics, and kind of the difference in the representation of the two, the different kinds of features that are focused on with the two. Because primates are both vertebrates and mammals, understand the evolutionary history of these two groups, knowing kind of what favored vertebrate radiation, and then knowing the advantages that mammals had over any reptiles that survived that mass extinction event at the end of the Mesozoic, is useful in understanding kind of why have so many mammals today, and be able to recognize and discuss these theoretical perspectives relating to the contemporary understanding of macroevolutionary processes. This includes the concepts of both species and speciation. This is crucial to interpreting long-term aspects of our evolutionary history, and this is critical when we're looking at the process of fossilization and how species how we interpret fossil specimens in this kind of greater overarching scheme of species versus genus. So, lastly, understand this species and genus designation, how they are used, and what their underlying biological significance is.